So last class we had talked about the sliding filament model where the actin is anchored at one end of each myofilament at a position in striated muscle tissue called the Z-line, shown here and here. And then movement of actin also moves the Z-line because they're anchored together. And then as actin moves past myosin, it drags the Z-line towards the myosin. Both actin myofilaments are being pulled inwards, so here and then contraction occurs. All right, so muscle contraction we had talked about involves the interaction of those two myofilaments, the actin, shown here, and the myosin. Here we are seeing the myosin head as well as the whole myosin filament. So during this process, the sliding filament model of muscle contraction, the sarcomere, which is the region of overlapping actin and myosin filaments, it shortens in response to muscle stimulation. That sarcomere, it repeats itself along the length of skeletal and cardiac muscle fibers. So we saw those Z lines, which will move towards each other when the thin actin myofilament moves past the thicker myosin filament. Okay, and then we will see that contraction occur. And we do require ATP for this to occur. And next we'll be looking at the role of calcium ions in contraction. So when a muscle is relaxed, its myosin heads are raised and ready, but they are unable to bind to actin. So the attachment sites for the myosin heads on the actin are blocked by a protein called tropomyosin, which we can see here in the blue. And that tropomyosin has to be removed out of the way for a muscle to contract. So there's a protein called troponin, shown here, and it binds to the tropomyosin. So the troponin and the tropomyosin, they form a complex that is regulated by the calcium ion concentration of the sarcoplasm. Okay, so that's again the cytoplasm of muscle fiber. So when the calcium ion concentration in the sarcoplasm is low, tropomyosin inhibits myosin binding and the muscle is relaxed. When the calcium ion concentration is raised, it will then allow calcium, it will bind to troponin. So this troponin-tropomyosin complex, it shifts away from the attachment sites for the myosin heads on the actin. So here we can see they've kind of shifted off of the those attachment sites. And the myosin heads are now able to attach to actin, and then using the ATP energy, they can move the actin filament to shorten the myofibril. All right, so in this diagram, first diagram here, we can see it's at rest. The muscle is at rest. That tropomyosin, it's blocking the myosin binding sites. It's tightly wound around it. Without actin's ability to bind with myosin, muscle contraction cannot occur. So here we have those calcium ions here, and they bond to another protein molecule called a troponin, which is part of the actin myofilament. The resulting complex now will reposition the tropomyosin. It'll expose the myosin binding sites of actin, shown here, and now the myosin heads can bind to the actin and contraction can now occur. So calcium ions are very important and they have this role in muscle contraction. Energy for muscle contraction. So the body's muscles require a continuous supply of energy in order to maintain their function. When the energy demands for muscle contraction exceed the reservoir of stored energy, the ability to control movements may become impaired to the point that normal voluntary coordination of even simple tasks like running are impossible. We know that muscle contraction, it requires a continuous supply of energy in the form of ATP. There's three main sources of ATP. There's creatine phosphate. It may also be referred to as the phosphophagin system. There's aerobic respiration of glucose, and fermentation of glucose, which we had saw in the previous unit. It was also called anaerobic respiration, or the lactic acid system. Creatine phosphate breakdown and as well as fermentation, they're both anaerobic, so oxygen is not present. Creatine phosphate breakdown is used first. It's a way to acquire ATP before oxygen starts to enter the mitochondria. Aerobic cellular respiration can only occur if oxygen is available. So if exercise is too vigorous that oxygen can't be delivered fast enough to the working muscles, then fermentation occurs. Fermentation causes an oxygen deficit. All right, so in this diagram, creatine phosphate builds up and it's stored in a resting muscle, and shown here. So for the muscle to contract, it needs to acquire ATP. So in figure A, when the muscle starts contracting, it breaks down stored creatine, the stored creatine phosphate. That's going to generate some ATP that's used immediately. To continue contracting, the muscle carries out aerobic cellular respiration as long as there's enough oxygen available. When the oxygen gets used up, the muscles can then carry out fermentation for a limited period of time, as we saw when we were doing the cellular respiration unit. We can see the glucose, the pyruvate. Oxygen is available, it will go to aerobic cellular respiration. If it's not available, it goes to fermentation. All right, and we can see the products here. From fermentation, we have some ATP and lactate. From aerobic cellular respiration, we have ATP, carbon dioxide, as well as water. All right, and then muscle can contract, and the ATP gets broken down to ADP and a phosphate molecule. All right, so regardless of what source of ATP is used, when ATP is consumed during muscle contraction, the result is an ADP molecule and a phosphate molecule. These will then get recycled back into cellular respiration to create more ATP. So during prolonged endurance exercise, such as running a marathon, a depletion of muscle sugar stores, a decrease in blood glucose, dehydration, an increase in body temperature, they can all contribute to fatigue. And during an intense muscular activity, lactic acid is formed through glycolysis and that lactic acid fermentation. The hydrogen ions from lactic acid, they decrease the pH of the muscle, and it may inhibit metabolic processes and thereby can decrease the work output. All right, so this is just a summary chart of the energy source, creatine phosphate, aerobic cellular respiration, and fermentation. So creatine phosphate, it's anaerobic. It doesn't require oxygen. It's occurring within the muscle myofilaments. The initial reactant required is creatine phosphate. The final products are creatine and ATP. And the relative amounts of ATP product, it's high, but for a short period of time. Aerobic cellular respiration requires oxygen. It's occurring in the mitochondria of the muscle cells. The initial reactants are glucose or fatty acids. The final products are carbon dioxide, water, and ATP, and there's a high amount of ATP produced. During fermentation, it's anaerobic, so it doesn't require oxygen. It's occurring in the cytoplasm of the muscle cells. Initial reactants are glucose or fatty acids. Final product is lactate, also called lactic acid, plus ATP, but the amount of ATP produced is quite low. All right, so in this lesson of 10.1, we looked at cardiac muscle, smooth muscle, and striated muscle. We looked at the structures in muscles and how muscle contraction occurs. Also, what's the relationship between energy used by muscle cells and heat production? So in this summary then, the three types of muscles, the skeletal, smooth, and cardiac, they have specific characteristics. We know that the smooth muscle, it's found in the digestive tract, in the blood vessels, and it pushes substances along a tract. The skeletal muscle functions in our voluntary movement. Cardiac muscle is found in the heart. And all these muscles, they contract according to that sliding filament model of actin and myosin interaction. Okay, then we talked about the sources of ATP, and in the event that aerobic cellular respiration can no longer proceed. So we know that as ATP is broken down to release energy for muscle contractions, heat is also released as a byproduct. So let's take a look at some questions here. What is the role of calcium in muscle contraction? So we know that calcium, calcium ions, they will bind to troponin, blocking myosin from the active sites on actin. And question 2, what is the correct order for the following statements when describing a muscle contraction? So the correct order is number 3, the myosin head is attached to actin. The myosin head flexes, advancing the actin filament. The myosin head releases and unflexes, and that's powered by ATP. And then lastly, the myosin reattaches to actin farther along the fiber. All right, so we already watched those first 2 videos. So I'm just going to pause the recording here and we'll play these last 2 videos on the sliding filament theory.